Hi, I'm David Salzman, the founder and CEO of sRNAlytics, and we're developing the next generation of biomarkers to diagnose and treat dementia. Dementia is an epidemic affecting 45 million people worldwide, and as the population ages, this number is set to grow to 135 million by 2050. Spending in the dementia space has exceeded $600 billion per year, making it the largest of any disease area. And the challenge is that dementia is actually a blanket term used to describe a number of diseases that at their very earliest presentations look identical to a doctor. And because there's no access to the disease tissue for biopsy, just like there is in cancer, patients often undergo over a dozen tests over a 4-year period of time just to reach a diagnosis that we know from retrospective studies are incorrect 25% of the time. This means that 1 in every 4 patients that's enrolled in a clinical trial doesn't even have the disease that the drug was designed to treat. This has contributed to an almost 100% failure rate for drugs in this space, costing companies over $1.5 billion and 10 years of time per failure. This is not normal. No other disease experiences failures like this, and in fact, it's so bad that it prompted the FDA to come out with new guidance mandating drug developers to incorporate new biomarkers into their clinical trials that can enroll patients with early-stage dementia. However, no such test exists on the market and has left drug developers scrambling to find a solution. I recognized this need 4 years ago when I was working as a scientist at Biogen, and I left my job there to pursue a vision to turn small RNAs into biomarkers. I have 15 years of experience in the small RNA biomarker space, and I started grad school working on them when there were only 6 labs in the entire world studying them. My graduate thesis focused on uncovering the mechanisms by which small RNAs are formed and how they function in our cells biologically. And my fellowship at the Yale Cancer Center focused on turning these small RNAs into diagnostics for cancer, to not only diagnose disease, but also to direct medical therapies for patients coming into our clinic. What we learned from my work is that all of the tissues in our body have a unique small RNA classifier associated with it that can actually do things like tell you what tissue of origin the disease is originating in. And we also understand that diseases originating in our brain will send out unique signals throughout our body that we can then detect in a liquid biopsy and turn into the diagnostic molecules that the FDA is calling for. So to do this, we've developed an end-to-end discovery pipeline to identify small RNA biomarkers that we call sRNAtypes. However, we see 2 addressable markets here. Number 1, to fill the need for the clinical diagnostics that the FDA is calling for, and also a second market to test pharmacodynamics that we know will be immediately opened as soon as this first diagnostic need is met. And we are currently simultaneously developing these products to meet both the current and future needs. The big difference is in our data. A lot of companies right now are working in small RNAs. However, they're limited to a list of 2,588 genes. And at sRNAlytics, we've developed a novel computational algorithm to process, sort, and analyze the small RNA sequencing data, giving us high visibility resolution to sequences at the single nucleotide level. What this does is it's expanded our discovery set to over 250,000 unique targetable entities, giving us over 100 times power to discover compared to our competitors. To develop proof of concept, we analyzed small RNA sequencing data from a group of patients harboring a genetic form of dementia. And we were able to produce this extraordinary heat map. This is a game changer, and no other method in the world can produce data like this. What you're looking at is a sRNAtype for Huntington's disease. And in this heat map, every column represents a patient, and every row represents a biomarker. And as you can see, all of the small RNA biomarkers in green are only present in the Huntington's patients, and they are never found in the healthy controls. Similarly, all of the biomarkers that you see in red are only found in the healthy people and are never found in the diseased. And these are also indicative of all of the normal biology that is lost when a patient gets sick. What we've built is an extraordinarily powerful classification tool that companies can use to derive unambiguous yes-no information to direct patient decisions in clinical trials. However, we understand that in order to make our technology scalable, we have to come up with a non-sequencing based solution. So we turn to RT-qPCR, and we do this because it's a fast, cheap, easy to use test that can be deployed throughout the entire world. And we develop these targeted assays against our sequences, and we validated our computational data. We have 6 patents filed in our space. We have 1 PCT filed on our method to develop sRNAtypes, as well as 5 additional patent applications spanning different subtypes of dementia. And as our products move through the FDA approval process, we unlock larger and larger targetable markets. Having filed our patents, having filed our patents, we can now begin selling our sRNAtypes into a $33 million addressable market. And what we're doing right now is we are forming a coalition of researchers, neurologists, and clinicians around Dr. Richard Myers, our scientific advisory board member. And he's going to the FDA to file a letter of support to use our biomarkers in clinical trials. These letters of support are a mechanism that the FDA uses to gain support for investigational use of new biomarkers in clinical trials. And when we've generated a big enough use base, we will go back to them and file a biomarker qualification package, at which point we will become the de facto enrollment marker for every patient presenting with dementia. Over time, we can file for a 510(k) approval, at which point we will unlock the entire market of everyone over the age of 65, which is valued at $138 billion. We are currently validating our biomarkers with 4 academic collaborators, and we've been in talks with a number of pharmaceutical partners who want to incorporate our sRNAtypes into their drug development programs. We have an exceptional team. I have over 15 years of experience spanning both academia and industry. Nathan, our CTO, brings 15 years of experience in computation and big data analytics and drives all of our computational-based discovery. Alan, our COO, has over 45 years of experience bringing high-tech innovations from concept to worldwide implementation. And Sean Melville leads our business group with over 5 years of experience in the diagnostic space. When I started sRNAlytics, I had a vision to make us the world leader in small RNAs. So I built an advisory board in accordance with that. Dr. Frank Slack is a Harvard professor and discovered the first human microRNA. And Dr. Richard Myers cloned the Huntington's gene and has discovered small RNAs in Huntington's disease as well as Parkinson's. And Dr. Joanne Wiedis is a pioneer in the use of small RNA biomarkers in cancer. We are sRNAlytics, and we're raising $4 million to help sRNAtype dementia. Thank you.